Hmm. Oh. I got that. Okay. That's what we were working on last time. Let me show you. Oh god. Oh no. That was a crash. Okay, we were working on that last time. Prove that you're not AI. Okay, I'm going to do that right now. Here we go. My stream deck will stop working. Okay. Weird. I can't. What is going on? Edit, remove. Dang. And this pic right here, right now, me right here, I am. Okay, let's do this again. Why can't this work? It's so frustrating. What are you doing? Dude, my freaking mouse isn't working. That's exactly how AI would respond. Exactly. You're not fooling anyone, Buster. There we go. Okay, here we go. This. Okay, here we go. Okay. All right, guys. AI. I'm AI. Wow, Stars, you think I'm AI? No way. My stream deck's not working. Dude, I don't know why this thing has not worked. I want to get a new one. It's very buggy. It just doesn't work sometimes. The whole thing's just doesn't work. It's very frustrating. Okay. I'm not AI.
What are you talking about? What, me? You think I'm AI? You think I'm AI? Are you out of your mind? I'm just a normal guy. What do you mean? Pretty good. Did the voice change? I was working on that. Did the voice change? Was that good? Was it good? It's supposed to sound like demonic. Nobody's responding. Did my voice change or not? Satan on the South Park voice. Okay, is it off?
Is it off now? Shoot. Uh-oh. During the thing, did it turn off? Now it's off. Oh, okay. Weird. Okay. I don't know why it should have turned off. Monster voice. Oh, here we go. Off. Toggle on. It's off now. Okay, here we go. Okay, one more time. Here we go. Oh, you think I'm AI? You think I'm AI?
What are you talking about? I'm not AI. I'm not AI at all. Are you out of your mind?
That was weird. What happened? What happened just now? That was crazy. Weird. That was super weird. I can't tell jokes. Do you know any jokes about microbiology? No. Honestly, I'm so bad at jokes. I always I can never remember them. It's impossible for me to yeah, I can never do it. Cool. I just know your mom jokes, yeah. Mostly mom jokes kind of guy, you know. New layout. Add item. Dynamic camera grid. Oh, click present. Select the window. Select PDF, extra camera. Yeah. Okay, present. Share screen. Thanks, guys. Share screen. Aha, nice. Okay. Can I make it? Oh. Oh, this is interesting. Ah. Weird. Trying to share my screen. Who do I like better? Sorry, Galviness. I know. I'm sorry. I'm sorry. I missed you too. I know. I just needed to get some stuff going. And just try to think. Yeah, the thing is I've invested so much money into the whole co-host idea, and it's crazy. It really doesn't work on any other platform, any other thing. So I've like invested a lot of time and money into that. So it's like devastating that like there could be a problem. But I don't think like I never have had any problems with like my science stream. Um it's always like with my political stuff, and I think that's way better. Um so yeah. Oh, Brittany, the changes in the algorithm and stuff. I mean, like people are still gonna be co-hosting, you know? Nothing, Michelle. I mean, my point is that I've like invested a lot of time and money into making the TikTok co-host thing. That's where my whole new show is centered around, the science show. This is kind of like the standard kind of like podcast world setup that I have. I've got stuff going on, all the stuff, so it's not that big of a deal. It's very bad, STLA. Yeah, we've been following that since like there was 10 cases. So it's incredibly bad. Yeah, it's really bad. There's no way to put it other than that. It's bad. Yeah, so I'm just kind of like having fun setting it up. Whoa, what the? Whoa. Yeah, it's really bad. It's really bad, guys. I mean, the it's insane. No, I mean it's just the problem is just the like the For You page and stuff has been really rough on people. So like they just haven't been scrolling and views are down because of that. People are like uninstalling and they're like they're not like super jazzed about it. So it's tough. It's tough. But at the end of the day, I think it's okay. I think, oh, this is cool. Oh, okay. Oh, guys, big news. Okay, guys, do this if you haven't already. Um, this, this, okay. This is the awards. We're gonna do this. We're doing this. So please, um, vote.drgregshow.com. We're gonna do that by the end of the month. I have a lot of stuff. I have stuff to go on Friday, and then so yeah, and then we'll do it soon. Have you tried a small boost? No, it's just like like literally um like just the people are just not excited about using TT right now. So it's like that's the problem. Yeah, call me Bob. That's never really, I mean, I kind of always am posting all my videos, all my shorts right before I go live, so that it's just kind of like what I do, so that kind of happens. Yeah, just because like that's how that what time I normally do that stuff in. Yeah, exactly, Heather. Exactly. Yeah, so that was my thing. Like, it seems like in the last week, it's just been kind of like insane. So I didn't want to like push on water. You know what I mean? Absolutely, Hazel. It'll be better than the Oscars. Absolutely, dude. There's also like some cool. Hey, what's up? There's also some chill music in the background for this other website. What in the world? What in the world? I don't know. It's so funny. It was funny. One of my first times I ever made my videos on YT. It was like someone was like, "I would love to listen to this philosophy of science discussion, but there's a jazz song that's like just going nonstop in the background, and I can't hear anything." Yeah, it's like what? So what's the plan? Are you just gonna wait, hoping to stop seeing rubbish? Yeah, Ness. Yeah, that's kind of my point. Is that I kind of have to because um, I'm like invested into the invested into the co-host idea. So I've done some other things to kind of like broaden, and I talked to okay, John, welcome in, dude. I'm glad you're here. Um, I I talked to some people, and dude, the numbers that they bring in are just insane. Um, and so I feel like I need to push on that more. Um, the red site, like I need to. No, it is Ness, but like just it's more about the for you page. That's my whole problem. Co-hosting was fine. It's the for you page and people actually getting my stream on when they're swiping. So that's why I just took 2 days off to like, you know, let it die down. I don't think that there's gonna be less enthusiasm for using like TikTok in general. Um, although there could be, you know. I think but in general, like I think I'm okay with it. Hmm. Hmm. Oh. Oh, that's one thing. That's interesting. This and then this. Can I do that together? I'm just getting everything like all set up for both things, you know. I gotta like I again. There was a point in time where I basically only did TikTok, and then I was like, oh, I'm gonna branch out, and then I basically said like, no, I need to double down into TikTok, and then now this nonsense is happening, and I'm just like, maybe I do. Oh, maybe I do something where I can do both, and that's what I'm setting up right now in the top. That's what we're working on right now. I just don't have time to like do this in the day. If that makes any sense, guys. So we're just like co-working. It's a co-working stream. Dude, maybe I do put it up there. Yeah, sell boxes of supplements. So it is what it is. I'll figure it out. I mean, it's more of just like a security thing. You know what I mean? That's the problem. It's just a security thing. Do I like sell the cameras that I got? That's the problem. It's just a security thing. Kind of start over again, or going at like more of a classic kind of like podcast show again, which would work on other stuff really well, or do I just like keep investing into TT? I mean, that's the problem. That's my question.
Well, I mean, the question is also like how do you need to invest it right now?
I mean, it's more of like a fun thing.
Or should
for
you
me.
just invest time?
It's more of like a fun thing for me, where it's like making something like different and new, and like having an idea that I think is cool. That's like more of what it is.
And then the specific type of video I'm talking about, first of all, like your regular content videos, you probably don't want to put those on right before you go live. You actually want to stagger those through the day.
Mm-hmm.
What you put on right before you go live is, hey, it's Dr. Greg here, and I'm going to go live on topics XYZ, and we're going to talk about this buzzword and that buzzword and this hashtag and that hashtag, and you fill the thing full of hashtags and buzzwords, and it actually does drive people into your live. It gets you on the FYP.
Yeah, that's interesting.
I mean, it's not as effective as it used to be, but it does work.
Interesting. Yeah, I just kind of...
And especially...
Go for it.
Yeah, I mean, it works. And you put it on... You put it on like about a half an hour before you're going to go live. Half an hour to 15 minutes before you go live.
Okay. Yeah. That's
and you can make them funny, you can make them silly, but it's sort of like you have to do that like super I'm selling you voice with it. Yeah, and Ness says, and do it in the suit. Put your suit on.
Exactly.
And say, hi, have you ever talked to a microbiologist about COVID-19? Well, now's your chance. Anything, right?
Yeah.
And then you say, ask me questions, bring your controversial questions, all of that kind of stuff. Works.
Okay. Yeah, I've never done that before. I mean, I would do it like hours before when I didn't realize, I didn't even know what I was doing. But yeah, that's an interesting idea.
Well, no, I mean, like, this is separate from your regular content videos.
Yeah.
This is...
Hi, Hannah. Yeah, I'll figure it out. Yeah, it's honestly bigger. I need to figure out the like branching into the red and the purple site and the blue site. I like their viewership was, and I've seen that in a lot of other places. It's just like it's wild. I mean, the super chats and stuff there. It's like it's crazy. What's up?
Well, the co-streaming has worked for a lot of people where they're streaming on more than one platform at the same time.
Yeah, the problem is like doing the science show. So like here, I know exactly what I'm going to do and how to do this, but I don't know how to put the science show onto the restream, because basically like I need to have the video of the person that I'm talking to somehow captured in the browser. And it's like not so simple. Yeah.
And also,
Thank you,
here's my
Hannah.
little dog.
There you go. There you go. That's crazy, Ness, that you didn't have any US content for a couple days. That's crazy. That is wild.
I know, Hannah. He does need a late night talk show like Jimmy Fallon.
That would be good. Yeah. We'll have to see. I'm trying to figure it out right now, and I'm like kind of, yeah. I'm just kind of, it's tough. I don't know why it's not working. Did you guys see creators from the rest of the world? Not specifically, Ness. I didn't see US for the few days until I got a VPN and set it to San Francisco. Wow, that is wild.
Well, Greg, that is another thing you can try is use a VPN and see if it pushes you out to different people.
That'd be tough. The quality would be so bad. That'd be the worst
Using
part
a
about it.
VPN?
Mm-hmm.
No.
Yeah, for
No.
streaming. Yeah. You'd need like really good solid internet connections, and if you're just being rerouted like 2 or 3 times for VPNs, it's
Try Proton VPN. Do you know this guy Various who just joined went to CERN?
Oh,
and he
cool.
worked at CERN.
Oh, nice.
It's very cool.
Nice, dude. Hell yeah.
Maybe you need to show off more jujitsu moves.
Yeah, maybe. No, yeah. I mean, that's like it'd be fun to do more different types of content for sure. I mean, that's what that's what I want to do in general. I was thinking about that, but I want to do
more jujitsu.
Well, I mean, just like still do like one night on the open mat, and then do one night with the podcast, like once a week. I want to record like a podcast for like once a week at least, so that people can listen to it.
Yeah.
And then do, so that's 2 nights a week, and then take a night off, and then also, so that's 3, and then 4 nights a week trying to do the show or something. It just takes a lot more effort in order to do the show. It's tough.
Yeah. I think maybe attracting people with more talk about the activism we're doing, getting more people pushed to the Discord could help.
Yeah. The Discord, I think, definitely. I think, but the problem is like talking about those things like limits your FYP. That's the thing. Like mentioning any off TikTok app hurts your algorithm reach. So
does it? Are you sure?
Yeah. The TikTok representative talked to me about that. That's why I was pushing Service Plus. That's why I was like pushing that so hard, because it was like the only way that I could get. They were like, we would do the opposite. You'd be pushed on the FYP for using Service Plus. So, but it never really took off. Like, that's the thing, is that even
it like didn't really work for everybody.
Yeah, exactly. So that's the thing, is like it doesn't. People can't actually use it. Like, you can't see it, and it's like, then it's like, well, this isn't really working. Yeah, like how many leads did I get?
Yeah, Hazel, TikTok is a little bit like a bad relationship. It's like I wish
Yeah,
I,
Heather.
I could
So
click you.
I understand why Ness like didn't get the button because she's like in a different country. Maybe it's
Mhm.
U.S. only, but Heather, you live in the U.S., right? So that's why I'm like confused. I would have thought that you would have gotten it.
Well, and it helps if everybody has updated their own version of TikTok to the most recent, and if they haven't, it may. I don't know. Maybe you should. Maybe you should just start selling custom outfits for the boo boos.
There you go.
That seems to work pretty well.
There you
The
go.
algorithm. Your shop needs to sell those fleece line tights.
Let's see. Let's see how the responses have been. Sheets.
But in other news, I am absolutely flooded with pro-vax stuff on the Book of Face.
Oh, really?
So that's
There you
been
go.
very interesting.
That's nice. That's cool.
Yeah, I've been aggregating it out to everybody.
Yeah,
And
yeah,
I have
exactly,
some
Ruben.
ding-
That's why I
I
took.
have some ding dong friends.
That's
on-
why I
on the
took the
Book
last
of
2 days
Face.
off. Yeah, because it was just like things are like being weird. Nice. Okay. Yeah, dude. You get 18 responses right now for the TikTok show awards. Guys, if you're just coming in, vote. Take a screenshot. No, you don't have to, Ness. No, you don't have to. I'm everywhere. That's the thing, is that like I'm literally everywhere. You know, like that's this.
Yeah,
we're everywhere.
I'm not getting your vids on the face place, but I do get your stuff on the the Tubi place.
Oh, nice. Ruben, no, I haven't. I took the last 2 days off, so I took the. Yeah, it's all good, dude. You got me just as I started streaming again, so you're good.
I thought you were going to take tonight off too, and I was getting upset. I was like, "Fuck, what am I going to do? I'm going to have to like put a movie on or something."
Was
Who
it
wants
actual
that?
on the Book of Face? Yeah, I mean, people are.
Hazel, I'm getting factual because I'm getting like World Health Organization. I'm getting like everybody's state and county health department, because they're trying to say, hey, don't listen to the feds. They're being idiots. Listen to us.
Yeah.
Like the image I received, like that I am displaying, because I've been doing a lot of, like when they're posting good memes, I'm like grabbing them. I also got this one.
Oh wow, it's only $8 a month. Okay, maybe we do that then. Maybe you do X. Dude, Discord's amazing. Don't you dare, Ness. Don't you dare. It was, Ruben. So they were purchased like 3 months ago by they transferred ownership because it was technically banned. ByteDance is a Chinese company, and they banned ownership of it, and they had to let people in with new ownership. And basically, what happened was they transferred ownership over to Oracle. Oh, gotcha, Ness. Gotcha. Yeah, I needed to do that. How does the TikTok shop work now? I mean, it all goes through the creators anyway. All right, calling about. Thanks for coming up. If someone else wants to come in, appreciate you. Appreciate with the idea with the video. I'm going to do that going forward for sure. All right, thank you. What's up, dude?
Hey. Oh, can you hear me?
Yeah, can you hear me?
Do I hear? Yeah. Sorry, is this a debate channel? I just saw the
Sure.
thing of like conservatives or subs, so I wasn't sure if it was a debate channel or just someone shit posting.
Okay, sure. What's up? What do you think?
Are you a progressive Democrat?
I'm a scientist.
Oh, okay. Do you mostly talk about scientific topics? I wasn't sure if you're a science.
No,
or
we talk about
politics guy.
science and politics, whatever.
Okay. Let me pick a topic that's an intersection of both, since that's your expertise.
Okay. I mean, whatever you wanted to talk about, man. It's okay. You don't have to, it doesn't have to be science based.
Yeah, I would say you're in microbiology, correct?
Yeah, I have a microbiology PhD.
Okay. Would you say most of the applications for microbiology are for pharmaceutical?
No. I mean, there's with the expansion of microbiology in the last 10 years through next generation sequencing, we kind of know that their microbes are everywhere. So it's like a lot of it is actually in plant root microbiomes, like for example. I mean, there's just and in like renewable and sustainable energy production too. So it basically microbes do three things really well, which is like creating new materials, creating food, and creating fuel. Like medicine is like a part of that. They're basically like 3D printers for proteins and stuff like that. But that's how they're used as microbial workhorses for sure, for medical purposes. Yeah.
Do you kind of view my understanding is there's a lot of hype about nanorobotics in the future?
I actually did my PhD with nanotechnology. I'm actually the pioneer of using nanotechnology with algae. So
okay, perfect. So I'm interested because there's a lot of hype among like the technological accelerationist groups.
Mhm.
I don't know much about the biological space, but they're getting pretty hyped up on nanotechnology.
Mhm.
And I guess high level, what's been the breakthrough that's getting people excited? Has there been some sort of breakthrough that's allowing us to create new materials and more advanced nanorobotics, or is it just traditional hype? Like we've had these techniques for a while.
There has been recent breakthroughs in the last like 3 years, and the ability for like batteries, like in order to like batteries and circuits, and that kind of thing. It is there has been like something, but really, like in general, like nanotechnologies existed for a really long time. Like a big misconception that I had was that it was like just recently invented because of some new, you know, physics fabrication process or chemistry. In reality, basically nanotechnology is just the functionalization of nanoparticles. And nanoparticles are anything lower than 100 nanometers, which is a lot of things. So, and we've been like functionalizing nanoparticles for 50 years. It's not that. Yeah, I mean, and nanoparticles exist in nature. So, like for example, a single-walled carbon nanotubes, which is what I used in my PhD, they get emitted from forest fires from the tree trunks. So, it's everywhere.
Is most nanotechnology? I know this is a very vague and open-ended question, but
Yeah, okay,
stick with me.
you're fine.
Are we mostly fabricating them via biological methods, or are we doing it via some sort of like industrial chemical process for building these nanorobots?
Like all chemical, like no biological. There is some this is actually an interesting question that I had coming into it, because I was a microbiology PhD. So I was like, oh, there's actually these Antarctic bacteria that actually make nanoparticles in order to protect themselves from the UV radiation of the sun. So, and then you can actually change the emission pattern of those nanoparticles based off of what you feed those bacteria. So you could change it based off of giving like more things like cobalt or something like that. And so it's actually, you can tune it a little bit, but compared to like taking a nanoparticle and then just functionalizing it with a coating and then binding something to that coating, it's so much more like quick and simple for chemistry to do it than biology. It's just everyone just does chemistry.
Gotcha. So would you say the techniques are similar to the techniques we use, for example, like chip fabrication in terms of scaling up? Or we're just using these really advanced chemical processes and very refined inputs to mass fabricate these nano particles?
From what I understand in the chip manufacturing that's happening, like in order to make the GPUs and stuff like that we use, it's like a super advanced mechanical engineering feat, and this is more like chemistry. I would say it's more chemical engineering than like necessarily like advanced mechanical engineering.
Okay, gotcha. So you're still mostly based inside a chemical engineering perspective in terms of your scaling up.
Yeah,
You're not okay. So you're using like traditional chemical engineering vats when you're productionalizing these techniques in industry. It's not like you're using
there's a, like, for example, so basically the process that we needed to do was like we got carboxylated single-walled carbon nanotubes, and then there's single-walled carbon nanotubes, like the chirality is already set, the length of them is long, and then you basically take that carboxyl group and then you add kind of like a coating to it, and then once you add that coating to it, it could be anything, but we did an amine, polyethyleneamine, which is basically kind of like a plastic, and then bound DNA to that. So that, and then that kind of like was the chassis that you're using. So, but the actual like chemistry methods in order to interact with this stuff is like simple, man. We're talking like filters, we're talking like centrifugation, it's you're just titrations of chemistry. So it's like, it's not like you're using these advanced machinery or anything to work with them. It's just like a centrifuge and some filters. It's pretty wild.
Oh, so essentially you're telling me that it's not like that you need to make these massive 10-20 billion dollar investments to scale up the production of these nano machines, or what's the proper term? Is it nanorobotics or just
okay, so there's several types. So nanotechnology is the umbrella term, and then within there's like DNA nanotechnology. So the idea that like the DNA can actually form structures itself, so you can actually create these kind of like basically tobacco mosaic virus is an example of this, where like there's like these proteins that kind of like come into a chiral form and it creates this long tube, and then the tobacco mosaic virus, and in between that the RNA is like stored there. But in the same way, you can actually take like building blocks of DNA that are going to be building and actually binding to one another. You can build that up so that you could have like a huge DNA tube basically, and the tube is made up of pieces of DNA that you would want to deliver. So like the idea is that you would have like, you're delivering like some type of gene or something like that, but they're big. They're really big. So that's, and then DNA nano robotics are basically little, those are like giving you a function, you're giving you a signal or something, remotely or not, into like that system. So it's a little bit different. There's also like nanotech.
Is there actually transmission? Like, can you actually transmit via radio signal to these nanorobotics?
Yeah. The problem is like batteries. So how long can they actually withstand and withhold the charge? So that's a big thing that was just being worked on. If you're interested in this stuff, it sounds like you are. Strano Michael Strano, Strano. He's like the head of like where all this stuff is being. He's incredible. He's incredible. It's where it's being
MIT.
debated. Yep. Yeah.
Interesting. Okay, so nanorobotics is real. We've achieved it, and we're actually able to communicate and build nanoscale compute.
On the very simple functions, yeah.
Wow.
Yeah. So, but it's not like, but they have limited capabilities, and like it's like very simple functions, that kind of stuff.
Just like move up, move down, left, right.
Yeah. What's better, like what's honestly like more interesting, I think, is kind of like, you can connect them and keep them connected through like a patch or something like that. You can, you don't have to like Strano had a paper where they actually did, this is like, God, 10 years ago now, did they did a bunch of nano stuff. Like they basically put a bunch of carbon nanotubes in like nails into a board, and then put that onto the brain of this mice, and then they were able to kind of like sense what was happening inside of the brain of the mice, which is pretty wild.
And I'm assuming that it's not even invasive because it's nanotechnology. So you could essentially just do delivery via pill.
Well, the patch is going to be very invasive, right? Because you're literally laying a patch on it. If you had like free form single, like delivery of nanotechnology, then you're basically looking at how are you applying it, and basically you can paint it on, you can spray it on, for at least that's how we did with the plants. That's what we're working with that. But the delivery mechanism is kind of like in that's being developed. That the goal would be to have something like that, but that's not happening in mammals right now. That's like, it's completely that's not happening.
Gotcha. And by a patch, are you saying like a dermal patch, or are we talking like
kind of like a Band-Aid kind of thing? You just kind of like put it on, and then that's like what it is. I mean, the idea is to have that. The idea would be to, especially if you're sensing things, if you have single-walled carbon nanotubes and you have it set up in order to sense for the detection of a single protein, the question is like, if you had that protein, you found that protein and it gave off a signal, like, would you be able to see it? And that's kind of the problem. So the patch creates an ability to kind of have all your sensors all ready right there, so that when it does happen, you just boom, it's good. It's right there. It's more concentrated, and you're more likely to actually like sense and see those, and where to look for if you have the camera, right? So you can have like a camera trained to that area, and then you can see that, like, okay, this is right here, because you basically have an excitation and an emission for everything. So if you need to excite something with a laser, and then once that that changes into the emission, but once it bounces off the chemistry and all that kind of stuff, then it's going to emit into a certain wavelength, and then you need to be able to capture that with a camera. And if you just have like free floating, in an organism, if you just have them free floating, it's going to be hard to see. So like this right here, actually, like right here, right, this is actually my own microscopy images that I made for my PhD. So this right here, these are dye bound to DNA. And then this right here is just the chloroplast autofluorescence. So this is just the internal structure of the algae. And then this is the overlay. So basically where the magenta and the cyan overlap is going to be white, and you can basically measure that. So there's like little white spots that you can kind of like see. So it's like a way for, you can actually see this stuff like go in. That's like, that is like the cool thing for a lot of this like nanotechnology stuff, is sensing what's happening, knowing what's happening.
Yeah. Is the main benefit that essentially understand what's going on at a nano scale without using an electron microscope.
So you can know what's going on, and then also they have unique capabilities of like dealing with and lowering the amount of reactive oxygen species. So reactive oxygen species are created when you have a damage, when you have damage coming. Basically your stress, abiotic stress, all that kind of stuff is going to be, wow, Ness, thank you so much. Thank you. Appreciate you. It's going to cause at low amounts, reactive oxygen species lets your body know like something's going on. We need to signal this is like really important. But at high levels, it can be detrimental and it can be toxic. So what nanotechnology allows you to do is actually kind of like scavenge and find those reactive oxygen species, and then basically take those oxygens, those free radicals, out of the biological system, and allow the biological system to be kind of like purged of them. So it has a pretty unique capability to do that. And yeah, that's what we've done. It's really cool. We did it. The lab did it, and for coral reefs, figuring out like the bleaching of coral reefs, like dealing with that by applying nanotechnology. We also allowed plants to grow in like more salty conditions than they could normally by just like, again, letting it deal with like that reactive oxygen species by purging those, taking it away. So yeah, there's a lot of applications.
Yeah. And would you say one of the big bottlenecks for improving is going to be just like basic material science, or do you see other major bottlenecks in the advancement of the field?
It's really going to be, and what I tried to do in my PhD was trying to like kind of like merge synthetic biology and nanotechnology, because the real bottleneck is kind of like delivery of these synthetic biology tools. Basically we have like these proteins, we have these capabilities, these molecular tools, but how do we know where it's going to go into the cell? Right now that we use adeno-associated viruses, AAVs, in order to basically look onto like specific cells that we're going to enter into, and we use different ways of like figuring out like exactly what cell it is in order to deliver the exact
so
stuff.
You're using viral techniques to essentially carry the payload.
So yeah, if you think about it, this right here, like this, what I'm delivering with the DNA through the carbon nanotube is actually has the same function as a DNA virus. A DNA virus is going to be trying to infiltrate the DNA, and it's going to try to, put its DNA into the chloroplast in the same way that my nanotechnology is. So it actually, there's like it kind of like a I mean, it's kind of like, it's a subsect of nanotechnology called viral nanotechnology. So actually using like viruses and nanotechnology as a way to deliver kind of, and it's, people were like, let's design nanotechnology that looks like viruses, or like that has the same function and that kind of stuff. But now we've kind of like realized that like with CRISPR kind of changed the game. You can kind of just it's tough. I mean, CRISPR is big, so it's hard to just like deliver it. So you do need to get it in some way. It can be hard to like package all of it into an adenovirus. So you have to kind of like do like some weird stuff. But yeah, that's the limitation basically, is like how are you going to get all your DNA based stuff into the cell, and how do you know which cell it is? So the idea is using nanotechnology in order to kind of like help that decision process.
Oh, okay. So would you say we're at the, like, let's say we have a payload, right? Whether it's I'm assuming that you could put a biological payload in a nanomachine, or you can put nanomachines inside a biological payload.
Mhm.
Is it easier to manipulate nanomachines at this point in terms of the radio transmission, or is it better to just rely on like natural biological processes for, like, I'm assuming the main application is like attacking certain types of cells, right? Especially for things like cancer and
Yeah, like
anything on the medical side.
yeah, right now, the one that David R. Liu, David R. Liu, like they do adenovirus, adeno associated viruses for delivering their mRNA lipid nano like based stuff. So it's all yeah, it's basically you can't fit a protein inside of a virus, but you can fit some RNA inside of it, and that's kind of like that's what they're doing. So
Gotcha.
it still relies on the system in order to express it. That can be kind of difficult, but there's ways of getting around that, but you get the specificity with the virus because it's an associated virus, and AAVs you can tune it really well to like to attack just that one specific cancer cell, which is good, which is very good.
Gotcha. And help me understand too, with nanotechnology, I don't know my skills quite well, but can you get it as large as it can carry entire cells, or is typically nanotechnology at the subcellular level?
Oh yeah, you're looking at like DNA and RNA. So you are delivering the DNA, you're delivering the RNA, and then that makes the tool. So you're delivering the instruction manuals into the cells, and then the cells are making the tools inside the cells.
Gotcha.
Basically, the CRISPR tools themselves it's hard to get them into the cell. It's bigger than an ion channel, and you're not going to be able to get a protein that large just into a cell. You'd have to tear the cell. You'd have to do some type of chemical lysis, like in order to make it more porous, stuff like that, which you can do, but like at the same time, it's much better to, especially if you're going to do in situ work, which is mean that you're going to do the work inside the cell, like inside, you need to deliver it.
Gotcha. Okay.
Mhm.
And this is going to sound very uneducated, but
That's fine.
I know nothing about this field.
All good. All good.
Are we at the point where we're manipulating at the atomic level, or is it purely nothing smaller than at the RNA level?
We're doing now. We're like, so David R. Liu had the breakthrough of the year last year for like manipulating the DNA of an unborn person, changing the DNA to rid the DNA of the disease, and then that person was born without the disease. They've also been able to, with the same technology with CRISPR, basically changed the genome of a person that had childhood leukemia. She was born with childhood leukemia. Bone marrow transplant failed. Chemotherapy failed. She had 2 years to live. Within 6 months of administration of this medicine, she was cancer free, and she's been cancer free for 2 years. So it's like I mean, we're at the point where we're actually changing the genomes of, it's called the somatic genome. So like in my testes is going to have a different genome than the skin cells of my body. You differentiate so many times through stem cells and that kind of stuff. They're completely different. So it's whether so the kid with childhood leukemia, their somatic cells were changed, but that doesn't mean that their heredity, the eggs that she had, were still going to have that disease if they had that. So we're at the level, like, which it's pretty crazy though. I mean, you'd think that there would be so many cells in a human, in a fully born human, there'd be no way to make that many changes so easily in all these cells in order to cure the person of a cancer. You would think that there would be some cells left over so that the cells that the cancer could just come back, and that's not the case, which is incredible.
So, are these nanomachines self-replicating? They can't be.
No,
Right? Like
no,
you got to
no,
inject
no.
a ton of these nanomachines to hit every single cell, right?
The AAVs, so the adeno, it's an adeno associated virus. Yeah, I mean, you basically, you're injecting it, and it's very specifically finding the cell, and then even more specifically finding that part of the genome, and then on top of that, they are also using DNA sequencing to make sure that there is no, like, it's called off-target effects. So off-target effects is when the CRISPR finds something that looks similar and cuts and does damage when it shouldn't. The original Staphylococcus one that they found does a lot more off-target effects, but David R. Liu actually created a new one, a new type of CRISPR that never been happening in nature before, and that it makes it super specific and creates only one change at a time. So it actually base pair edits from an A to a G or a C to an A, like that kind of stuff. So it's super small, edited, precise. It's incredible. Like David R. Liu will definitely get a Nobel Prize. I can think of 2 already. He should get legitimately. I genuinely think he's going to be the first person in history to get 3. He's an incredible. He's a genius. Like, he legitimately is a genius.
Yeah, I'm looking up him right now.
Yeah, he's awesome.
Are these nanomachines? I mean, how much of the genetic code are we talking? Are we talking just 1 or 2 base pairs? Or are we talking like thousands?
Really good question. Really good question. So right now they're focusing on diseases that are only a couple base pair edits. So they're just changing those, and those actually a lot of diseases are like that. It's not like a small amount that are like that. And then on top of that, like he's created, so the bespoke. Okay, so the problem is basically you have a lot of different types of like genes in your body for things, and they're duplicated. You have a gene not in just one location, but all across all these different ones, and they might have different mutations on each of those new locations. So if you could have one disease, but you could have a ton of different types that are just specific to you. So basically every single time we create one of these personalized medicines, we're going to have to make a bespoke one for you personally. And that's what they did with the kid, by the way. They literally created an all new bespoke, medicine that just for that baby had all the different mutations all accounted for, and they fixed them all. Another thing that he just did 2 months ago. I mean, we're talking 2 Nobel Prize for what David R. Liu's already done.
Honestly, I'm in the David R. Liu fan club now.
He's
I've never
incredible.
Heard of this guy, but
he's incredible. That he just created something new that genuinely I think is probably the biggest advancement since antibiotics, which is a crazy thing to say, but I think it's warranted. Basically, he's created a way in his mind, and they actually demonstrated it with a paper that it's called like sequence agnostic disease curing. So basically they're creating one medicine that can cure a ton of different diseases, like 100 different diseases at a single time, which is unbelievable. So because the biggest problem with this stuff is going to be accessibility. So if every single person that needs to have a disease cured is going to be $1 million, it's not going to be accessible to anyone. So therefore nobody's going to use it except for rich nations, and rich nations are going to have decreased medical costs, while poorer nations are going to have more disease and increased medical costs now. So, to have one medicine that can actually cure 100 different diseases, and it's basically by messing with the tRNA. Basically, he created it's genius. I mean, the molecular biology is a little difficult to give an analogy for, but basically he's created a way that's like, hey, you don't know what a table looks like in your instruction manual. You basically, you've come home from IKEA and you are putting together your table, and the instructions, one of the instruction pages is gone, and you're like, I can't do this. I can't make this table. So David R. Liu has basically created a IKEA, like no matter where you bought your table, IKEA or not, here's the instruction manual for how to make a table. And you look at your table, you look at this thing, and it's like, oh, I can build this table now. So that's basically what he's done, but it's done with tRNAs and basically the ways that to override stop codons, because a lot of times how diseases are made is basically you have a premature stop codon. So the protein is being made, and then all of a sudden there's a stop in the middle of it, and you can't make that protein anymore. You can't make that protein, which makes the disease, right? So now he's created this. He was like, okay, so we just create this medicine based off of the tRNA that can just be read through right here. So then that means that all these diseases that have this tRNA, right, can all be cured. So it's like, it's genius. It's just because he looked in so much of biotech is just like stealing ideas from nature and then weaponizing it basically how we want it. And he's done that. He did that with this tRNA thing, and he's demonstrated it. So it's going to human trials right now. It's genius.
So quick thing. So this nano robot can essentially read DNA, do a local compute, and then based off the DNA that it's reading, do some sort of programmatic solution in terms of editing. It's like, okay, I've detected these 30 pairs. So it's just now compute, say what I should do, and then do it.
So just to be specific, it's not a nano robot. It's a virus that they genetically engineered in order to deliver CRISPR. And CRISPR is a bacterial defense mechanism. So it creates from DNA, it creates this protein. This protein inside the cell goes through and matches and finds a match for the designated site that it's been shown or instructed to do based off of the DNA that exists. It binds to that DNA, it changes that DNA sequence, and that's how it works. So it's actually DNA. Sorry, it's like nanorobotics is just a separate field, and like it is doing cool stuff. Strano is like doing cool stuff with
but this is purely biological.
Exactly. That is the differentiation. Exactly.
I had no idea that we had that level of compute that could be performed at a biological level.
Exactly.
Where essentially you can say, I'm going to receive an input, make a decision on how to do it, and then do it all over with
dude, genes. Holy crap. Thank you, man. Thank you so much. That's so kind of you. Dude, it's crazy. It is unbelievable, man. Like it's like science fiction. Like it like legitimately is like science fiction, and people if people knew like what we could do. it's remarkable, dude. It's amazing.
And I understand why a lot of these accelerationists are getting hyped on it now. Are we doing it just via brute force random experimentation, or is there a lot of simulation that goes into creating these like miracle genes that can physically do compute and then start editing customized to any case?
So this is another stroke of genius that he had, and this is why I'm telling you, the most scientists would be lucky to have one thing that would be, a genius thing, but he's like done it with like 5 different ones. So he did it through with directed evolution. So he created these things based off of and it was actually crazy. In 2017, he gave a seminar at my university talking about, and his seminar was about how he was making through directed evolution, because Francis Arnold just got the Nobel Prize for directed evolution the year before. And he was talking about how he's using directed evolution for making these new CRISPR medicines. And I was like, oh, this is so cool. Everyone's using directed evolution. This is amazing. So that was like my original thing to try to use like directed evolution, my PhD and stuff, and it didn't work out, but whatever. And but it's basically they use we can synthesize now like millions and millions and millions of different variants, and we basically create an experiment where evolution pushes the product that we need and creates it that way.
So the idea is that you create a bunch of random molecules,
Mm-hmm.
you then deploy them, and then you selectively breed to see which one gets closer to your end goal.
Yeah, you tie like the fitness of like the fitness of the organism somehow tied to the product that you want. So it's like you'd somehow make it so that if it did make an edit in the right mutation, then it creates some type of fitness advantage. Like now it's accrued antibiotic resistance when it didn't have it before. So now like the one that had the best capabilities to make it the fastest would get the strongest antibiotic resistance. So then it would proliferate inside the media, and then you could tell by its sequencing which sequence is the highest number, and then you say, oh, okay, like this is the best enzyme that does this.
It seems maybe I'm just biased because I'm in the computer science space. But,
Nice.
It sounds like they're using machine learning techniques just with biological.
Yeah. Frances Arnold does that too. Frances Arnold had an incredible paper. She did. It's a lot of deep learning because there's basically more data than we can go through ourselves, like as scientists, and deep learning can create those correlations that we wouldn't be able to understand. So that it's been used for like specifically in directed evolution for a long time, like 10 years, I think.
Gotcha. So they're using those techniques to assign like, okay, this one's slightly more fit, to assign the fitness score.
Yeah, exactly. And then, but then also like, tell me what is like, what could be the difference with it? Why is this one different? What's going on with this? Like that makes it so like this, like, is there any other than like just this score that we have right here? We already know that. Like, tell me an associated thing that I wouldn't be able to know. And it's like telling us more and more about it.
Wow. Okay.
Yeah. Frances Arnold has a paper about that. Frances, she's incredible, dude. She's at Caltech, really good. She's another one of my like role models as a scientist.
And are you essentially sequencing the entire? Okay, I want to make sure I use the proper term. What do you call the biological payload that's able to do this? Do you just call it? Are they considered proteins? Are they considered Okay, they're viruses. Okay.
Yeah, the adeno-associated viruses are viruses. That's the carrier of it, and then yeah.
So you're using viruses that have the CRISPR capability to then edit the genome of every individual cell. And the idea is that the virus can self-replicate and then just carry forward the CRISPR gene that's then editing your individual genes in every cell.
The virus isn't replicating. The virus, yeah, you don't need it to. You don't want it to.
So the virus doesn't. Okay, so the virus doesn't affect the cell.
It is just to exactly. It's just to do it, like to get into the cell. Like that's it.
Oh, okay. So essentially you will have one virus for every cell that you're trying to edit. Or probably like 2 or 3 viruses, because not everyone's going to hit.
Mm-hmm.
Okay. Gotcha. So we don't have to worry about self-replicating inside of the human host, probably because there's some issues with that in terms of health,
that would be it's a huge, like, ethically that creates a secondary effect, and then we don't need it. Once the goal is the entry into the the cell, and really like specifically we wanted to pick out the right cell. Like that is the big thing.
Okay, gotcha. And I know nothing about virology, but we can also custom code the virus to hit the particular cells, and that's a well-established field. Like there's not a lot of
for certain viruses, we've worked with them like a really long time, and we know how to work with them really well.
Wow. Okay. So essentially, it's not that difficult to generalize that specific. Well, I guess they're not species, but it's not hard to generalize and hijack a virus genome to then say target this cell instead.
In some viruses, yeah. I mean, it's a very small percentage of the viruses that exist. Like it's I mean, the microbiology is the final frontier of science on Earth. I mean, you have 99% of microbes that have never been sequenced or cultured in the lab. So there's like so much out there, I mean, that we don't really understand or know or worked with. So but they're out there, and viruses are even, I think that stats like there's more viruses on Earth than there are stars in the universe. So it's like they are everywhere. It's crazy.
And help me understand too, are you guys doing a pretty good job of documenting all of the different microorganisms and all the viruses, or is there still a ton that are undocumented?
Tons still undocumented, because the next generation sequencing just came out, which allowed us to sequence microbial communities for the first time. Before that, Sanger sequencing required us to have a single cell like inoculum. So basically only one strain at a time. And microbes are kind of like their microbial communities are basically like houses of cards. You remove one, the whole house of cards is gonna fall down. So we need to sequence the entire community, and we just started being able to do that. I don't know how many viruses are in the trapped ice, but there are ones that our body has never grown accustomed to, which is definitely scary. Hopefully, like none of them can bind to human cells, because if they can, we're screwed.
Is that because they can cause rapid mutation?
No, it's because our bodies have never even dealt with them. So like SARS-CoV-2 was a novel virus because our bodies hadn't seen the spike protein that went into their cells, so we didn't have an immune response to it. These viruses, not only would our bodies not have any antibodies towards, but also the scientists, we would never have worked with them before. We have no idea how to work with them. A coronavirus we work with all the time. That's nothing big. But like a new virus that we'd never isolated before, we've never worked with, we don't know how to genetically modify. That's a huge problem. It's a huge problem.
Gotcha. And because we don't even know how, essentially, since we don't even know all the viruses that exist, like the number of viruses we probably don't even know, let alone we haven't even sequenced them.
And like even though these viruses are going to be from a time when humans aren't even around, right? It doesn't matter when it's basically like a directed evolution experiment, right? Where you have all these different mutations with all these viral genomes, and then you're just going to get one or however many that hits a human cell, and then you're off, and it could be awful.
And because of the simplicity, I'm assuming the mutation rates are higher with viruses.
Yes and no. So like they do need some type of like mutation rates, but you don't want to hyper mutate to the point where you actually like mutate away from your host. So like it's called, you can kind of think about it as a valley. Like it's like you want to try to mutate your way out, but you could mutate to the point where like you're not able to catch on to anything.
Gotcha. And are we, now that we're at the point where we're starting to, in a sense, we're directing our own mutations using these new techniques where we're using these
humans are?
Yes, essentially we're essentially doing human induced mutation using these techniques where we're editing our genes.
Okay.
Are we going to get to the point where we could also apply these techniques to viruses to speed up and create even customized viruses? I'm assuming.
I mean, interestingly enough, David R. Liu in the directed evolution experiment that I was talking to you about, he actually utilized viruses in order to inject new DNA into the ongoing directed evolution experiment so that he could actually because normally what you start with is like a fixed pool. It's called the mutant library of like all the different mutants that you have. But then like getting new mutants into the system can be kind of difficult. But he actually utilized viruses in order to deliver new DNA into the directed evolution experiment, which is why he's probably should. I think that he delivered the Nobel Prize probably for that, just for that alone, and then another one for this.
So viruses infecting viruses.
Viruses infecting cells, and the cells were creating the CRISPR in order to edit the genomes of the cells in order to make them more fit.
Oh, wait. So we use cells to create the CRISPR
Exactly.
RNA. Okay.
Exactly. And he used viruses to deliver new bits of DNA in order to test them out, because you want to try out all the permutations that you can. That's the idea. I mean, you want evolution to figure out what the best mutation is. And in order to do that, at different points, you need new mutations. It's not always like a linear thing. It's not like always you want the best one at any given time, because like the best one is just, it's not the best possible. It's the one that's just good enough at that time. And sometimes like a mutation that was really bad before, and now it has another mutation far away on that protein, and like it unlocks some exterior hindrance that it had before, and then now that protein has like hyper capabilities, and now the active sites are completely different. So you actually kind of want to reintroduce some mutations and kind of always have that fresh material kind of going.
Yeah. And are we still relying on individual cells to produce the RNA, or are we going to get to a point where we can just use like a computational simulation and then assemble it ourselves because we have nanomachines that can just build it better than a cell could?
Yeah. So like, I mean, AI and like neural networks are being used right now in order to generate hypotheses that still need to be tested. So like AlphaFold is a good example of this, where it's like, for basic proteins and like simple peptides and stuff like that, you can actually have to you can have some ideas on like what might happen, but you still need to test it inside of like a biological system. It got the Nobel Prize a couple years ago, and I thought it was like a little bit premature because of that. It's like, it generates hypotheses. It doesn't generate like new medicines like instantly.
Yeah, the simulation technology is too crude to actually do it. Yeah. You need to actually run the experiment and then see with your own eyes.
Like maybe in the future. I mean, that's honestly what we need to happen. That's what the current administration's banking on, by the way. I mean, RFK, like they're banning all manimal like testing. So, and no animal testing on any medicines now, which is, other than humans, I guess they're keeping animal human trials. But yeah, they're banking on basically these computer models filling that gap, which is, I feel like that's very wild.
Do you think that a lot of the scientists using animal techniques are just going to migrate because all the other countries will keep it?
I can't imagine. Let's just say I can't imagine nobody doing animal testing. Like, if you had banned animal testing 100%, like you would just have more humans being harmed at the testing stage, because you would have, even though all your due diligence, you'd have all the right tests, all the models that you would ever want, but there still is, like, I mean, in biology, we call it the biological black box of, like, everything is set up, everything should work, but just something doesn't work. And it's a living system is so hard that you can't predict every single aspect of it. So I think what's going to happen is that more people are going to get harmed, basically. But I hope that it goes to other countries, basically what I'm saying. I hope it does.
And just so I understand, I'm assuming they can't ban private companies from using animal testing. It's
Yeah,
just,
of course they can.
we won't give federal grants.
No, I mean, they can ban it. They can.
Oh, so it's a ban for everyone, even private researchers, not just people reliant on grants.
In the US, yeah.
Holy shit.
Yeah, it's reckless. I mean, it's reckless. I mean, it's insane.
Is it to pander to, like, the animal rights groups, you think? Because of his, like, background?
Probably, like, yeah. I mean, he doesn't have, like, they don't have any respect for science or the scientific method and that kind of thing. So I would think so. Maybe that's the most plausible solution that I could think of.
Yeah, because it definitely feels like a bleeding heart, like, animal rights argument because I'm assuming it's for research and, like, for, like, products and vaccines and everything, right? Yeah.
Ironic, because he's saying that we need more research on vaccines, and yet he's banning research on vaccines. So I don't know. How are you supposed to do research on vaccines now?
And is that on the books or just proposed?
I thought that they're phasing it out. They're gonna start phasing it out right now. Like, I thought that was, like, the thing, but I don't know when. That's a good question. When it actually is, I think it's like by 2030. It's a good question.
Oh, okay.
RFK.
Because I'm assuming there's been no law passed, so this is just like an executive order type deal.
Yeah, I mean, HHS has a lot of power. As of is actively using it to phase out. I mean, we've already massively decreased the amount of, like, primates that we use for testing. It's kind of like I mean, it costs so much money, and it's so hard because you, like, to do anything, they have to have, like, live lives, like, they have to have, like, nice cages and stuff. It's very expensive, and it's very hard in order to do it.
Oh yeah, look, trust me, I'm on the science ethics. I'm on the extreme end of no matter the cost type deal. So
yeah, I tend to agree too, man. I think that, like, I mean, at least in the it's interesting, the human gene therapy side, that, like, what David Arlou is doing, there is no explicit ban in the United States on human gene editing. Some countries there are. The United States, we don't have one. Basically, every single, like, mutation or permeation of this medicine is has to be approved by the FDA. So that is the stop block from, like, some crazy, like, what's happened in China. So if you're not aware, like, in China in 2017,
CRISPR kids, with AIDS.
Yeah, so the problem with that was that it was the old CRISPR system, which had the insertion and deletion off-target effects, and it was like, yeah, it's just very reckless, very reckless. Rightly, the guy went to jail for rightly so.
Is it because the new? Well, my understanding, he was only editing the egg, right? The egg genome.
Yeah, he was editing out the part that the HIV virus, like, burrows into their genomes in order to, like, stop it from integrating into the genome of that person. So he was trying to make them functionally immune to HIV.
Yeah. But it wasn't like he could do it once the child was conceived. They could only do it at the pre-fertilization layer.
Yeah.
But with this, you can now edit genes post-fertilization.
Exactly. Yeah. Once the baby's been born, once the cells have been divided, everything. Hey Brian, what's up, man? That's the cool. That's one of the coolest parts about it. And like it means that like legitimately there is a world 10, 20 years from now where we go in, we get our genome sequenced, we figure out what's wrong with us. You go into your PCP and they just give you a shot, and like a bunch of diseases that you had in your genome that made you more prevalent to cancer rates or had problems here and there, you're cured of those. Like it's just gone.
Now, can we agree from a public relations perspective, we just won't call them vaccines, we'll just call it something different?
It's okay. I talk about vaccines all the time. We can talk about vaccine.
No, I'm just saying, but just from a, like, to me, this feels very, obviously it's the same techniques as vaccines because you're using viral payloads to, edit the genome, but
no, they're different.
like
So, I mean, it's gene therapy. So a vaccine is for inducing an immune response or an antibody response. So there's like gene therapy is using DNA and RNA in order to edit the genome of something. So it's different. They're both molecular biology, but they're distinctly different.
Oh, I always thought that the whole point with viruses is that they were actually manipulating the genes of the cellular host that it infected. That's not the case. It goes in, replicates, and then kills the cell and disperses.
So endogenous retroviruses do that, where they like sometimes they do like go in like and they integrate into the genome, but sometimes they don't.
Yeah, because I thought that was the whole thing with the C-19 vaccine, was that it was slightly modifying our cells.
No,
okay.
It doesn't. No.
Huh.
Yeah, it was like one paper like with a cell tissue culture, and they showed that there was like it was a bad test, and then it was like but basically all subsequent like analyses and testing and everything like showed that doesn't happen, and it's like the RNA doesn't like integrate into the DNA. It has to get into the nucleus, which is incredibly difficult. So yeah, it's a misinformation.
Wow. Okay. So,
but you could give yourself the SARS-CoV-2 antibodies by editing your T cells, like with CAR T cell therapy or something like that. I mean, that is possible. That would be a gene therapy, type of vaccine. But we don't do that. What we do is we give your body a small portion of the virus in order to train on. And basically, you're delivering RNA that creates a spike protein, and that spike protein is recognized by your body, trains on that, and then now it memorizes into the T and B cells about like how what to do in the presence of this antigen.
Gotcha. And I'm assuming we didn't use that technique because it was just the speed of delivering the technique.
We didn't use the CAR T.
Mm-hmm. So we didn't use that method as the inoculation because we just didn't have it in time.
It's like the CAR T stuff is used for like cancers and stuff like that. It's way easier and less experimental just to give someone a piece of antigen, which is like the piece of virus that they to train off of.
Oh, okay. So we didn't really use a different technique then if we just used an antigen. I thought that we did something very revolutionary with the C-19 virus.
So normally
old school, just giving people antigens.
So yeah, so vaccines historically have been like inactivated or live attenuated. So inactivated means that they were heat denatured or chemically changed just a little bit so that it's not like an actual living virus, but it's still there. It'd be like taking your glove off or something. So the glove is like not on your hand, but it's like it still resembles it. A live attenuated one is actually using directed evolution again in order to create a virus that is similar to the original virus in terms of like the the proteins on the outside so that your body can train off of it, but it doesn't actually kill the host anymore. So that's the thing. But it's an actual live virus that it doesn't kill the host though. So that's why some people can't have live attenuated viruses. But the thing is that the mRNA technology that we use with COVID vaccines, so basically it's a piece of RNA that creates the spike protein. So it's delivered into your body. It's RNA coated in lipid nanoparticles. And then you basically deliver that into these cells. And then your body creates a spike protein from that piece of RNA. And then your body learns off of that. So in a way, it's very similar, right? The antigen is the spike protein in the live virus, and the deactivated vaccines, those are the pieces of the virus. But what's really cool with the RNA is that we're actually making it inside of your body, or we're making that thing inside of your body. So and why that's really good, specifically for a pandemic, is that you can change the sequence more specifically. So you can create it to have the exact same outbreak strain that's happening right now. And during a pandemic, that's really, really good.
And help me understand this. Are we still using the traditional methods of put the mRNA inside of a virus, have the virus attach to the cell, or is it just pure mRNA that just knows how to
no.
cells?
That's the there was an adenovirus. There was an adeno-associated DNA virus, which is the Johnson & Johnson Johannes vaccines. That was the one that actually had a bunch of the people, like 24 people did pass away because of that. That's the only vaccine, by the way, that's had like deaths associated with it. And they pulled that one immediately. So that was like a new DNA technology that they tried, and it had problems. So that's what it was. Most vaccines are live attenuated or inactivated. That's what they all were basically until COVID.
Wow. Okay. And under this technique, there is no. I didn't quite catch it, so sorry. I'm asking the question again.
No, you're fine.
There is no. It's not using a virus to deliver the mRNA.
Correct.
Okay.
Correct. That's only with the David R. Liu technology, because they're delivering such a large amount of DNA, and it has to go into a specific cell. This stuff is like, we're just putting it into your muscles, and it just has lipids like around it, so that it doesn't immediately. It basically gets into the cell, but any cell. It doesn't matter what cell it is. It's a little different.
Gotcha. And I'm assuming the mRNA was way less complex in terms of like total pairs compared to what like David Liu's doing.
Oh yeah,
it's probably yeah.
Oh yeah, because you just have like a single spike protein that's being created. It's like not there's multiple. The medicine that he creates is like, there's a lot going on with it. There's a bigger protein. There's actually a guide RNA in there, which is it's a piece of RNA that actually directs the Cas9. There's another there's a couple of different parts of it.
Oh, so it's almost like a family of proteins.
Yeah, it's kind of associated. Yeah, a couple of them are associated with them. Yeah, exactly. But the spike protein from the COVID vaccine is just the spike protein.
Wow.
Mm-hmm.
And with the jumping back to essentially mainstream gene editing.
Sure.
There's no regulations blocking gene editing. FDA is the only one who can yay or nay it.
Yeah.
But you mentioned some other countries are anti-gene editing.
Yeah.
And I'm assuming it's because of essentially it edits the genes but inaccurately, and then that triggers a mutation which can trigger cancer. Was the main concern for slowing down gene editing technology?
There's just like a lot of apprehensive and like nature in some countries. Like it's just Europe, for example, is just against genetically modifying food. For example, they just have a genetically modified ban. So it's just
it's environmental GMO fear. Gotcha.
Partly, but it's very interesting because the United States has historically actually been very conservative, like cautious in like the things that we approve. There's a lot of experimental stuff that's allowed in a lot of other countries, but then still banned in the United States. So like that, we are kind of like the leader in genetically modifying organisms and stuff. Which, I mean, I'm a molecular biologist. I genetically modify things as my living. That is like what I do. So I'm biased, but there really hasn't been any negligible downside effects of like the dangers of genetic modified organisms like that have like played out, in my opinion. It doesn't exist.
For me, I'm not a PR person, but obviously there's been so much of like the PR moral panic with a lot of this technology. Like with everything. Do you think the solution is going to be just calling it something different and then just so people don't notice?
Yeah, I mean, people like the GMO label has been taken off of foods and they call it like a bioengineered product now. Yes, Lauren, we are. I do. I'm going to make a video about it today about it. Yeah. My family is participating. Yeah, I mean, I think so. I think it's important to talk about. I think it's important to like for scientists to talk about. I think it sucks. I think that it's very odd. Organic foods like actually require like more pesticides, like in order to be used. They have more runoff, they have more problems. I mean, because you're basically instead of having the pesticide be made inside the plant, at very small concentrations, you're putting a ton of pesticides on the outside of the plant, and it's just it leads to more runoff, more environmental harm, all that kind of stuff.
Well, also, like, ideally, if you can just GMO a seed to just be resistant, and I'm assuming you can get to a point where some of the plants are so GMO'd that the pests that eat them can't even eat it, right? Like essentially, it's
exactly
to them.
Yeah, exactly. That's the whole reason. I did see that Planet Peterson video on it, and it actually went on to his live, and I was talking to him about it yesterday. So yeah, thanks for telling me. Exactly. That's the whole idea, is that you kind of create the chemical defense that the plant needs in order to survive a pest.
Yeah, and I'm assuming too that one of the big problems is they're just going to argue of like, oh, which pesticide is more harmful, the naturally produced pesticide within the cell or the chemical pesticide that the farmer applies, and that's how they're going to run with it. And I'm going to guess there's also probably a decent amount of lobbying that goes in for like the chemical industry that produces the pesticide versus it being produced locally at the cell level.
For sure. Yeah, there's a lot of lobbying for sure.
But is there? Okay, and sorry, I'm rambling here.
You're fine.
This is just a new concept.
Yeah,
are we going to be using some of these gene editing techniques probably with plants first, and it's going to accelerate like plant biology research, or do you think most of it's going to be directed towards the human genome, human cells?
We're doing both at the same time. I mean, there was a thing called like the Green Revolution, which is when they started using molecular techniques in the 70s and 80s in order to kind of like increase plant productivity. So, we're doing it all.
It was more on the fertilizer side. I didn't realize it was GMOs even in the 70s.
Yeah, I mean that was specifically the fertilizer is basically the nitrification of the nitrogen gas in the atmosphere to nitrate, which makes it bioavailable. before we could do it chemically, it was the rate limiting step was bacteria, which is awful. And then so as soon as they were able to kind of like create that chemical reaction for bringing nitrogen from the atmosphere down into the biological form, then they can make a ton more fertilizer. But the problem is that the fertilizer basically runs off into the environment, has a ton of negative downstream stuff.
Gotcha. And with our current techniques, are we essentially just breeding plants that essentially need less carbon or need less nutrients, and they can just produce with less? Or is it more that they're actually producing it at the cellular level, the nutrients?
They're so you can create a new abilities for them to have an herbicide or a pesticide made inside of the plants, and then you can also enhance native plant functions. So you could increase photosynthetic rates, that kind of stuff, or the abiotic, stress response signaling pathway, kind of like what I was talking about before. So there's a couple different ways, like in order to increase, like it's called plant productivity per acre. That's like how you measure it. And I think there's a lot of different ways.
Gotcha. And I'm assuming too that they're trying to speed up the rate of cellular division in order to make it grow faster is also part of it.
Sure. Yeah, sure.
So grow faster. Therefore, you have a bigger plant within the season window.
Yeah. Yep. Exactly. You want more plants per acre. Exactly.
Okay. And with these techniques that we have, where we're now able to create generalized gene treatment for at the individual cell level,
mm-hmm.
and we can then generalize it to potentially the entire human population,
mm-hmm.
because it knows it can read, react, and then modify. Is there another frontier that's purely theoretical, or do you think this is the final technique and this will be able to generalize for all disease? In terms of, because like if you can edit your genes, right, that
mm-hmm.
ends pretty much all disease, right?
That would be for heritable ones, like the ones that you inherit from your parents. There's still diseases that are caused by external factors. So like cancers, like 80% of cancers are environmental. Environmental doesn't mean like necessarily from like ecosystems. It means from like smoking, for example, is an environmental thing, because it's like you exposed your lungs to an environment full of smoke, and now you have cancer. So it could be
mutates the cells.
I could see it like inherent, like we could like start curing these heritable ones over time, and maybe some of these larger disease ones, but it's going to be like really tough.
Gotcha. But maybe I'm out of my zone here, but right, let's say you're a smoker,
mm-hmm.
your cells mutate, they become cancerous. Couldn't you then create a generalized gene editing treatment that says, okay, let's attack only the cancer cells and then roll it back to pre-cancer in terms of the genome?
Yeah, theoretically.
Okay, but we haven't achieved that. We're simply modifying genes that pass down via reproduction, not genes that exist that are essentially specific to like lungs or liver. Or is that not the case? Because like you mentioned, it was a little girl with cancer with leukemia.
Yeah.
So that makes me think this isn't a genetic disease, or is leukemia genetic?
It was genetic, yeah.
Oh, okay. So it is the issue. I didn't realize that some cancers are just purely genetic. So
with 20%.
What? Yeah. Okay. What creates the difference where we can't edit diseases that are genetic? Is that because
there's too many?
Haven't sequenced every cancer cell?
There's too many of them. Like there's too many, like the changes that would be needed. It's like too big of a downstream impact. Like it's very rare for a cancer to like have like a couple of mutations. It's like that's just kind of tough. But that's why the David Arlou new stuff is pretty crazy.
Gotcha. And I'm assuming too, the idea of creating a generalized gene treatment is pretty new. It hasn't been tested widespread yet, correct?
Generalized gene treatment for humans?
Yeah. So you mentioned that, like, this technique with the little girl with leukemia only worked because they sequenced her individual genes and then created individualized treatment.
For sure.
But you mentioned there was another research where you can generalize it to the whole human population.
Yeah.
Has that been deployed, or is that more theoretical?
He just released that like 2 months ago. Like, I think so. It's going to be in human trials coming up. Yeah, it's pretty cool. I can't wait. That's going to be amazing.
Gotcha. And under that scenario, wouldn't that then be able to be? I know we're talking theoretical here, but you could then apply that to more complex issues where you have 5, 6, 7, 8, 9 mutations.
Yeah,
on an individual cell.
Yeah, if they all if they could all be fixed by the same tRNA thing, or maybe a combination of the both too. It's crazy. I mean, it's amazing.
Yeah. And my guess too, every cancer cell is genetically different, right? In the sense of, like, my cancer won't necessarily look like your cancer in terms of the mutations.
I believe so. I'm not 100%, but I believe so.
Gotcha. But I appreciate your time. And,
yeah, dude. Thanks for the conversation. I enjoyed it. I appreciate it.
Yeah, definitely. Sleep well.
Have a good one. All right, guys. I had jujitsu this morning. I've been doing 3 days straight of jujitsu in the morning, so I'm like exhausted right now. Yeah, I gotta go. Oh, it was a good throw. It was a good day though. Ooh. It was a good day. It was a good day. Got some subs. It was amazing. Thank you, Voice. That was really cool. Yeah, that was an awesome. Thank you, Joey, for coming in. I appreciate you, man. Thank you. Appreciate it. I will see you guys mañana, Thursday night. Yeah, I was tired. It's been good though. It's been really good. My jujitsu has been getting better. It's been way more fun. Dude, my wife's jujitsu, bro. Oh my gosh. Someone stop her. She's gotten so good. Mrs. Dr. G's jujitsu is so good, guys. It really is. I'm telling you, she has like the mind for it. She did 2-a-days yesterday, and then she did another one. She went to jujitsu 4 times this week, so she is tired. She wants us to not go Thursday night. Thursday, this today. I'm gonna go. We're gonna go. Ness wants to know if it's a rumor that the U.S. wants to block access to PubMed. Yeah, I don't know if that's. I mean, maybe. I'm not 100% sure. Oh, it's true. It's true, Voice. Muchas gracias. Buenas noches. All right, ladies and gentlemen, thank you for everything. I appreciate you guys so much. It's been fun. Hey, Corey. Hope everything's going good, man. I'm headed to bed. All right, guys. Thank you all. Love you guys. Thank you. Be kind to one another. Bye. Have a good one. Peace. All right. YouTube, Facebook, and X. I think this is gonna be the trio going forward. All right. See you guys. Bye.